good afternoon, and good evening. Today, we will deliberate about the application of gene therapy and its uses in the future. DNA, or deoxyribonucleic acid, is the hereditary material found in every human and almost of other organisms. Unique sequences of DNA make up of genes, some of which act as instructors for cells to make molecules called proteins. Genes which dictate our unique characteristics. Maybe you have a curly hair, straight hair, brown eyes, gray eyes, white skin, black skin, or brown skin. Our unique characteristics are required for us to develop and live. In turn, genetic changes, alterations, and mutations which may be inherited or acquired over one's life can directly cause disease or increase one's risk of developing disease. Now, let me introduce to you the gene therapy. Gene therapy is a technique that modifies a person's gene to treat or cure diseases, which involves delivering genetic material into patients' cells, can be used as a form of treatment in diseases with genetic component. Gene therapies can work by several mechanisms. Now, there are three approaches or mechanisms in gene therapy. First is replacing, inactivating, and introducing. First one is replacing. Replacing a disease-causing gene with a healthy copy of a gene or cell. Second is inactivating, or also known as knocking out a disease-causing gene that is not functioning properly. And last one is introducing. Introducing or giving a copy of a new or novel modified gene into the body to treat certain diseases. Now, how does gene therapy works? Let's take here an example of an illustration. Here is a new gene or a new genome, or also known as virus vector. Now, this virus vector is transmitted to the human body. Now, this virus vector is packaged or bind with the cell until it packaged with a vesicle into the cytoplasm. Now, when this virus vesicle is about to enter the nucleus, the vesicle breaks down and releasing the vector into the nucleus. And it's time to release the new set of cells injected into the nucleus. In scientific researches, the gene therapy poses a promising future into the human sphere. It cures a number of diseases such as severe combined immunodeficiencies or SCID, hemophilia, Parkinson's disease, cancer, HIV, and myriad of human diseases. Previously, we have talked about virus vectors. Now, what is these vectors? Vectors in the gene therapy is the delivery system or act as a vehicle that carry a set of cells, set of genomes, whose task is to deliver it to the target cell or to the defected cells. There are a lot of vectors used in scientific researches and scientific experiments, such as rectovirus, adenovirus, leptovirus, and alphavirus. But the most commonly used is retrovirus and adenovirus. In the world of gene therapy, there are two main types of gene therapy, such as germline gene therapy and somatic gene therapy. First is germline gene therapy. It is the introduction of genes into our reproductive cells, sperm cells for men and egg cells for women. Reproductive cells carry an individual's genetic information and pass it onto offspring. Hence, modifying the genetics of germline cells causes any changes that are made to be passed down to future generations and the coming generations. Now, how does germline gene therapy occurs? Let us take here an example of a scientific experiment of how this therapy can be done. First is the doctor get or removed the man's sperm that produced cells in which contain a defective gene. Now, these sperm cells with defective genes will be added to the novel or vector virus or therapeutic genes and modified genes to replace those defective ones. Next, these genes are put into mouse testes or into a rat until it become mature and start producing healthy sperm. Now, when these sperm are tested and are used to fertilize the egg cells in the testing area, the resulting embryos are placed into the woman's womb and then giving birth to a child free from the father's diseases. Now, here's another type of gene therapy, and it is called somatic gene therapy. Somatic gene therapy is the introduction of genes into the non-reproductive cells in our bodies. Now, what is non-reproductive cells? These non-reproductive cells are called somatic cells. These somatic cells are any types of cells in the human body that contain diploid. Now, what does that mean? It means they contain two sets of chromosomes that is inherited from both parents, from maternal and paternal. While somatic gene therapy in an individual in all their cells except for reproductive cells. Changes are therefore uninheritable. Whether or not genetic modifications can be inherited is critical in the understanding the scientific potential and risks of performing gene therapy in an embryo. Now, there are two types of somatic gene therapy, the in vivo and ex vivo. Now, let us contrast these two terms into its simplest idea. The first type of somatic gene therapy is the in vivo. It is the direct transmitting of genes inside the human body targeting the defective cells. While ex vivo is of course the outside alteration of the cell, then afterward is to transfer or inject it to the human body targeting the defective cells. Now, let's take here an example of an illustration that shows the difference or how it works, the in vivo and ex vivo somatic gene therapy. First is the in vivo, or also known as the direct delivery. Example of it is a therapeutic gene covered with and packaged into a delivery vehicle such as the retrovirus. Now, once the therapeutic gene is covered, it is ready to inject into the patient. Example of it is the liver cancer. Now, once this therapeutic gene is delivered, its target is to the liver cancer cells of patient. And the second type of somatic gene therapy is called ex vivo, or also known as cell-based delivery. Now, how does it work? An adult stem cells is extracted from the human body and is isolated and propagated into the laboratory room. Now, once it is isolated and propagated, in vitro differentiated stem cells. The same thing with the direct delivery or in vivo somatic gene therapy, a therapeutic gene covered and packaged into a delivery vehicle such as the retrovirus and is introduced into the cells. Now, once it is introduced, a genetically modified cells are ready to introduce into the human body. Now, it will target the defective cells and making a new set of cells. The application of gene therapy into the human society poses two major differences, such as advantages and disadvantages of gene therapy. First one is the advantage of gene therapy. Gene therapy gives a chance to a normal life to a newborn baby with genetic diseases, gives hope, a healthy life to cancer patients. For certain diseases, that do not have any cure except gene therapy, it could save many lives. It gives hope to the human who are in health of medicine, cure and treatment, fighting for life and fighting for survival. Yes, indeed, gene therapy has come into a whole new world, giving a better place for every human. However, the gene therapy poses disadvantages matters, such as the genetic testing, screening, and research in finding the availability of certain gene is very controversy. The cost is very high and the patient might need an insurance to cover the treatment. Many increased rate of abortion if prenatal test regarding baby with genetic disease is done. And even the gene therapy poses number of risks such as the way the vector is being delivered and transmitted into the human body. Yes, indeed, gene therapy poses two major consequences in the human society. It can be good or ethical, or it can be bad or evil. Now, the uses of gene therapy in the future will be discussed by Irene Nunez, Antoinette Lagilias, and Jenna May Sumagang. This is Ian James C. Capito. Thank you.